Perfecto. Hola a todos los que están ingresando en la sala, bienvenidos de vuelta. Hoy tenemos a Eva, así que voy a hacer una breve introducción de ella en inglés y nos va a dar su conferencia. So, hi to everyone once again. Today we have an amazing speaker here with us. And some months ago, when we started our evolutionary course, I made a short review of the evolutionary thought over time. And of course, I presented relevant protagonists such Lamarck, Kimura, Gould, Margulis, of course, Darwin. But when we get to the last slide, I presented researchers who are currently developing new aspects of the evolutionary theory, like Kevin Laland, Richard Lenski, and of course, Eva Jablonka. Eva is a professor in Tel Aviv University. She had a PhD in genetics. She wrote a lot of papers, but I think that most of you know who is Eva because 15 years ago, she wrote with Marion Lamb a famous book, Evolution in 4 Dimensions, a book that today is a classic with edition in many languages, a book that must be a read among the biology students. And Eva also wrote the famous article, Does Evolutionary Theory Needs a Rethink, together with Armin Motzek and other authors. And for all these reasons, I wrote several months ago, I wrote to Eva, asking her if she could give us a lecture to our students. And she replied, of course, and I couldn't believe it. And well, today is a reality. Eva is here with us to present a talk entitled The Extended Evolutionary View, the Epigenetics Perspective. So Eva, thanks again. It would be a pleasure to hear you.
Thank you very much. It's really a pleasure to be with you, although not physically, but virtually. But this is life today. So I will start and I will share the screen with you. And I will start with I'm going to talk about the extended evolutionary view, and particularly because it's a very, very big subject, I'm going to focus on the epigenetic perspective. So let's, this is going to be my talk. Now, I will explain this picture. This is the picture that we call the 5 mothers, but it's also the mothers are also fathers in a way. But anyway, these are routes of transmission of information from one generation to the next. And it can happen through the transmission of variations of DNA. It can happen through transmission of epigenetic profiles, different epigenetic profiles. It can happen through the transmission of all kinds of substances that affect the development of the embryo in utero. It can occur during early development through the transmission of food traces or immunoglobulins, and it can happen through the big the great through culture, which is really encompassing everything, all these routes. So there are many routes in which we can transmit information, and we know that this is not something new. I'm going to talk, my talk today, I owe it to a lot of people, in particular to Marion Lamb, with whom all these ideas and all this way of thinking was developed. I'm very grateful to my student and my colleagues, and to my friend and person who illustrates my work and helps me, Anna Zaligovsky. So in several years ago, in 2014, there was a paper in Nature that asked the question, does evolutionary theory need a rethink? So do we really need to make any fundamental change in the way that we think about evolution? Or are the things that we have discovered during the last 20 years needing only small adjustments in the existing theory? And 2 views were put forward in this article. One said yes, urgently, and the other said no, all is well. So I am one of the people who think that the yes urgently view is correct. And I want to try and convince you and talk and tell you why I think so. I mean, I don't really want to try and convince you. I'm not a lawyer, I'm a scientist. I want to put forward what I think and how I think, and why I think that there are very, very good reasons for a kind of gestalt switch in the way that we think about evolution. So the view that is the traditional view was developed during the 20th century and was crystallized during a long period, really. But it is, and although it has changed, it is still dominant today. I will not read all this long quotation here, but I will read parts of it. This is by Ernst Mayr, one of the architects of the modern synthesis. The modern synthesis is this view that married neo-Darwinism with all kinds of discoveries in genetics, population studies, systematics, paleontology, and so on. So it was a synthesis of classical Darwinism with new discoveries and new ideas that developed during the first more or less 50 years of the 20th century. And different people have different ways of putting dates on this process. It really was a process. It was quite a long process. What Mayr says is, it was in this years, 1936 to 1947, that biologists of the most diverse subdivisions of evolutionary biology and from various countries accepted two major conclusions. One, that evolution is gradual, being explicatory in terms of small genetic changes and recombination, and in terms of the ordering of this genetic variation by natural selection. And two, that by introducing the population concept, by considering species as reproductively isolated aggregates of population, we can explain all evolutionary phenomena in a manner that is consistent with known genetic mechanisms. And what this required, according to Mayr, is that the naturalist abandon their belief in soft inheritance, that is the inheritance of acquired characters, for example, and that experimentalists give up typological thinking, that is thinking about essence. Species have an essence. They have to think about species as a population with a norm of reaction and variations. And it led to a decline, in addition to that, in the concept of mutation pressure and its replacement by a heightened confidence in the power of natural selection, combined with a new realization of the immensity of genetic variation in natural populations. So certain things had to be abandoned in order for this view, the traditional view, now that is a dominant view for many, many years, 70 years more or less, which you can still find in textbook, to be accepted. So let us see what was rejected and what was marginalized when this view was taken. So first of all, non-genetic inheritance was excluded. There is no soft inheritance, Mayr told us. This doesn't exist. The only exception was human culture. The gene was the unit of heredity, and it was also the unit of evolution. This was the assumption. It was not questioned. No soft inheritance. So we don't have non-genetic inheritance. All inheritance is genetic. There is no soft inheritance, which means the belief in what is soft inheritance? It is the belief in a gradual change of the genetic hereditary material itself, either by use or disuse, or by some internal progressive tendencies, or through the direct effect of the environment. This is excluded. It doesn't exist, according to the modern synthesis. Third, niche construction, that is the construction of the environment by the organism through the cumulative construction of ecological legacies that are transmitted from one generation to the next. What the parents made and constructed is transmitted to the offspring, which maintain it, could develop it, or could change it somewhat, but basically maintain it and transmit it, and so on and so forth. This is recognized, but is marginalized in the modern synthesis. The assumption is that there are no special mechanisms that operate during macroevolution. Macroevolution is more and more and more microevolution. So macroevolution is an extrapolation of microevolution. And processes such as hybridization, polyploidization, and symbiogenesis, which can lead to saltational changes, are marginalized. Developmental constraints and affordances are recognized, but they are marginalized. They are not very important. The most important thing is natural selection on very small variations. The constraints, there are always enough variations to go around a constraint. And the pattern of evolution is tree-like. Web-like patterns are marginalized. They are, again, recognized. There are good biologists there, but they are marginalized. And hereditary variation is sought at the gene level. Genes are leaders in evolution. So you start your analysis with genetic variation, then you go to the phenotype, and then you go to selection. And there is a simple relationship between the genotype and the phenotype. So small changes in genotype lead to small changes in phenotype. And this is a very simple thing. Natural selection is the only direction-giving process in evolution. There may be other processes, but they don't give direction to evolution. Of course, the architects of the modern synthesis recognize that there are processes such as genetic drift and migration and even mutation pressure, although they marginalize that too. But they don't give direction to evolutionary change. The only thing that can give direction to evolutionary change is natural selection. These are the assumptions. All of these assumptions are challenged by the extended evolutionary synthesis, all of them. And I just want to put forward to you the core assumptions of the extended evolutionary synthesis before I go into details about, that relate to epigenetics. The core assumptions of the extended evolutionary synthesis are the following. First, that it is not only selection that gives natural selection that gives direction to evolution, but developmental processes share with selection the responsibility for direction and rate of evolution and the origin of adaptations. Second, that inheritance is not just genetic, but encompasses epigenetic, physiological, ecological, behavioral, and cultural processes. The third assumption of the extended evolutionary synthesis, which my colleagues and I have been putting forward and pushing hard for many, many years already. I started in the late 80s, is that developmental systems often facilitate integrated functional responses to mutation or environmental induction. I will come back to this, it's a very important point. The whole view is organism-centered rather than gene-centered. And there are additional processes such as developmental bias, a certain bias towards the direction that evolution can take because of developmental constraints and affordances, and ecological inheritance. This kind of processes help explain macroevolutionary patterns and contribute to the ability of lineages to evolve, to evolvability. These are the core assumptions of the extended evolutionary synthesis. Now, in the discussion between the people who think that we need an extended evolutionary synthesis today and the people who think that we don't need it, one of the advocates of those who said, we don't need it, it's okay, we are fine with what we have, with the modern synthesis, he responded to the suggestions to our suggestions by saying three major things. First of all, that a lot of the things that we have suggested are already incorporated within the modern synthesis. It's not new. It's already there. It has already become part of it. Second, that the list of subjects that need to be incorporated, those plasticity, a very, very strong focus on plasticity, on inclusive inheritance, that is, that inheritance is not only DNA inheritance, but there are many inheritance systems. The idea that niche construction is important and that developmental bias is important, they are just ad hoc. They're just collection of all kinds of things that we happen to like, but they're not really related to each other. People could have chosen other things. But this is our hobby. This is what we're working on. So that's why we decided that this is important. And three, that the data coming from the study of non-genetic inheritance, and especially epigenetics, is not strong enough to warrant a change. Now, this seems to suggest that if it was strong enough to warrant it, it would warrant change, right? So let us look at what we know about it, about what we know about non-genetic inheritance. Now, non-genetic inheritance is a very, very big subject. It's not just epigenetics, although molecular epigenetics is at the basis of all this, of all the changes that happen during development. It is simply the molecular correlate of developmental changes. So obviously it is part of it. But when we're thinking about non-genetic inheritance, we're thinking about the inheritance of traditions among animals, and also about the inheritance of legacies, of ecological legacies, and also about the inheritance of symbolic culture in humans. Now, I don't think that there is anybody who would say that the inheritance of culture in humans is not very important. And there are very few people who will say that animal traditions are not quite important for understanding of the evolution of animal behavior, although this is still not strong enough subject. But molecular epigenetics is the one that really makes people uncomfortable, because this touches a very sensitive nerve. It touches the basic molecular biology of heredity. And if there is good evidence and good reasons to think that epigenetic inheritance is important in evolution, then even Ray and his colleagues would agree that it warrants some kind of change. So that's why I'm going to focus on it. It's not the only reason. The other reason is that for the last, I don't know how many years already, but many, many years, I have done my PhD many years ago, beginning of the 80s, on epigenetic inheritance, on epigenetic mechanisms. Okay, so we're going back to this picture. So what we're going to focus on is on the second mother, mainly on the second mother, not only because, as we shall see, it is not possible to really make such neat dissociations. But we're going to look at the variations that are not variations in the DNA sequence, but are variations in all kinds of molecular factors that can be associated with DNA, but do not have to be physically associated with DNA. Okay, so this is a general picture just telling us something about the pathways of inheritance. We can have genetic inheritance that is transmitted through the gametes. We can have epigenetic inheritance, epigenetic variation that are also transmitted through the gametes, from gamete to gamete to gamete. We can have changes, we can have changes in the adult that go to the germline and affect the germline and then are transmitted from germline to germline. we can have transmission from one adult to another adult that bypass the germline altogether. So there are different routes of transmitting information. You can transmit information through gametes, but we don't have to transmit information through gametes, and there is a possibility of transmitted information from soma to germline, from the soma to the germline, from non-germline cells to germline cells, and then from germline to germline, or again, from soma to germline, from soma to germline, from soma to germline. And of course, from germline to soma, because the soma affects the development of the germ, the germline affects the development of the soma. Okay, so you probably know about what epigenetics is, and you know that, like many concepts, it has undergone all kinds of changes during its history. Its history is not very long, but it does have a history. And the person who coined the term epigenetics was Conrad Waddington, and it has a very broad sense. Epigenetic denotes the branch of biology which studies the causal interaction between genes and their products which bring the phenotype into being. This is very general. So what we have is the phenotype, we have the genotype, and the processes that intervene between the genotype and the phenotype, all this web of interaction that is in between, this is what he called the epigenotype. And epigenetics is the study of this epigenotype and the way that it changes dynamically all the time during development, and also when development never stops, but even when we reach the adult state, we have to maintain all the time the state. It's a dynamic system, so we have to maintain this system, and there is a dynamic going on in this great network that intervenes between the genes and the visible phenotype, the macroscopic phenotype. This is how he thought about it. So we are looking at this, at the way the genes are expressed and the gene networks are formed, cellular networks, developmental networks are formed, and this bring the phenotype into being. Now, in the modern sense, epigenetics is a much narrower term, and it is the study of developmental processes as we defined it in prokaryotes and eukaryotes. We is always Marian Lamb and me. Yes, we is not a royal we. It's okay. So it's the developmental processes in prokaryotes and eukaryotes that lead to persistent, and that's important, self-maintaining changes in the state of organism, the components of organism, or lineages of organisms. Again, this is a rather general and deliberately general kind of the most general that we can think of definition of modern epigenetics. Now, when we think about what happened during the, I think, well, it's difficult to put to pinpoint when exactly it happened, but definitely since the mid-60s onwards, we began to understand what epigenetic mechanisms are. These are the mechanisms of cell heredity, basically. And when we're talking about epigenetic mechanism, we're talking about cell memory and cell heredity. These are the two things that we are talking about. What do I mean? We have, let's say, when people usually talk about 200 cell types that we have. I mean, not including the brain, the many cell types in the brain. And probably this isn't anyway an underestimate. It depends how you measure how many cells you have in your body. But let's say we have 200 different cell types. Now, these cell types breed true. That is a skin cell when it divides, or if we are talking a stem cell for skin, when it divides, it gives rise to a skin cell and another stem cell. But it breeds true. It never, or very rarely will it become something else. Very rarely will it become a kidney cell, for example. A skin cell will not suddenly change its mode of differentiation and become a stem cell for a kidney or a kidney cell, or a differentiated kidney cell. This is very unlikely to happen. So they breed true, and they breed true in terms of their phenotype, in spite of the fact that the signals that led to them being skin cells are no longer present. So something that happened in the past, in the developmental past, is maintained, dynamically maintained through cell divisions. And the kind of mechanisms that maintain the particular phenotypic state of cells are the epigenetic mechanisms. Now, we have known that they exist for quite a long time, but we now understand some of them in quite good molecular detail. So again, I will not bore you because I'm sure that you have learned about some of them. The first one that was recognized, by the way, was DNA methylation. It's a modification of one of the nucleotides, the cytosine, and there is a dedicated machinery that can lead to the replications of patterns of methylation. Patterns of methylation can change through some environmental changes or developmental signals, and once they are changed, they can be inherited through a machinery which is to some logically similar to the machinery of DNA polymerase. It's a semi-conservative kind of replication of these patterns. We can have, so this is this methylation which was the one that was studied and identified already studied already in the 60s, identified, the mechanism was identified and thought about in the 70s, and afterwards it was shown which enzymes are involved and so on. So it's one of the set of mechanisms that change the way chromatin is organized. It's not the only one, because we have other changes which involves, for example, the changes in the histones around which the DNA is wound. So we can change chromatin by changing the patterns of acetylation, ubiquination, phosphorylation, methylation, and so on of these histones. And this changes through mechanism that we understand less well than the methylation mechanism, but we are beginning to understand them, can also be reproduced as the cells divide. And again, they are sensitive to developmental signals. Then we have something which is called self-sustaining metabolic loops, but basically it is the formation of loops with positive feedback, so that when you induce a change, it leads to the reestablishment of the same change again and again and again, even when the signal is no longer present. And this is something that we know from bacteria, exists in every cell under the sun. And it can lead to the inheritance of some of alternative states without changing the DNA. Of course, also the chromatin marks lead to the inheritance of alternative states of chromatin without changing the DNA sequence. We also have self-reconstructing three-dimensional structures such as prions. I hope you all know about prions, and we know that the same protein can have more than one conformation, and there are conformations that are self-templating. So once a particular conformation is in place, this conformation will lead to its own reproduction during cell division and during the formation of the protein. And we have the transmission of RNA, small and not so small RNA profiles. And there's again a specific machinery that can lead to once there is an induction of certain small RNAs, they can be reconstructed from one generation of cells to the next and can also reach the germline, which is very interesting. This, in some organism, it has been shown that small RNAs, which were induced in the soma, can find their way to the germline and can be inherited through the gametes. Now, we know quite a lot about these mechanisms, and of course, one cannot go into detail here. But what I want to say is there is no doubt that they exist. There's no doubt that we cannot understand development and differentiation and determination and morphogenesis without understanding this mechanisms. They are not the only mechanisms we have to understand, but they're crucial. And that they are important in development, there is no question. There is also no question today, I hope, that epigenetic variations in DNA methylation, in RNAs, in prions are transmitted from one generation to the next. How they are transmitted, with what fidelity, and so on, we'll come to this. But there is absolutely no question about it, and there is a huge amount of data. I did a review in 2009, and I stopped when we got to 102 cases. The reason I stopped 102 was it was a historical reason. I said, well, William Bateson, stopped at 102 when he was reviewing genetic variations in the beginning of the 20th century, when he was checking Mendel's laws. I stopped at 102 epigenetic variations. But this is nothing. I mean, today we know from work on Arabidopsis that we can have millions of variations which can be transmitted. So, this 102 cases were very nice at the time. Okay, so here, what do we know about the scope of fidelity of epigenetic inheritance? First of all, we know that unlike DNA replication, we have several different mechanisms, and in different species, not all mechanisms need to operate. We have some organisms, for example, that don't have DNA methylation, but they have, or very little. They have a little bit of DNA methylation, but they have very good chromatin histone-based variation inheritance. And the other thing that we know is that unlike DNA methylation, which can be replicated through a process of which is kind of replicative, in many cases what we have is not replication but reconstruction. So we can, for example, we can have a situation whereby we can delete all the DNA or most of the DNA methylation from a region, leave very, very little of it, and transmit some RNAs which are complementary to this region. And then during development, the old pattern will be reconstructed. So it's not like a photocopying machine. It's a reconstruction process, developmental reconstruction process, rather than replication. It's important to understand the difference. The other thing that we know about epigenetic inheritance is that it is ubiquitous. Everywhere we look for it, we find it. There is no organism on this planet that does not have epigenetic inheritance, including bacteria. Three, the fidelity of the transmission of epigenetic variations is very variable. It depends on the nature of the variant, of the epigenetic variant. It depends on the environmental conditions for how long, what was the history, how long this history was, how strong it was, and so on. And it depends on the taxon that we're looking at. It differs between taxa. We still don't know enough about it, by the way, but we have an idea that this is the case from the information that we do have. And if we induce changes, we know that stress can induce changes that can be transmitted from one generation to the next. So, and there is a lot of selection, and there is a lot of scope of selection at the epigenetic level itself. So we can have a lot of variation that is induced and inherited at the epigenetic level, and this can be selected. I mean, it can also, of course, undergo drift and all kinds of interesting other processes, but it can be induced and then inherited and selected. And for example, in plants, there was one classical study. They looked at the Arabidopsis thaliana, which is a small plant, and they looked at heritable, and the DNA was more or less the same in all the plants. It was an inbred, highly inbred line, pure line, and they looked at the pattern. They found more than 100,000 differentially methylated CG sites, methylation sites in the genomes. And when they looked at not at the sites themselves, but at the regions, they found several thousand differentially methylated regions. And the epimutation, the rate of change was about 4.5 times 10 to the minus 4, which is much higher than mutation rates, but it is low enough for selection to work on it. So at least, and in plants, we know that Arabidopsis is not that exceptional. Now, in other organisms, it varies, and in some organisms we know more, in some organisms we know less. I will go into it if you want, but this is just one example out of many. Now, I don't think that there is any doubt today that there is epigenetic inheritance. I don't think anybody doubts it anymore, except maybe some weirdos. But I don't think this is the problem. The question is, okay, so we have epigenetic inheritance. How do we treat it? Should we treat it as an extended kind of development, or should we treat it as something that has to do, or as evolution? Or do these distinctions actually begin to make become a little bit blurred? I will come back to the blurred question, because I do think that this dichotomy between development and evolution is getting somewhat blurred. So there are many, many evolutionary questions that we can ask. The first two, I'm not going to talk very much about them, because they don't worry the people terribly much. I mean, when I'm talking people, the people who are against the extended evolutionary synthesis, they say, yeah, well, that's part of what we have to think about, because we do recognize that epigenetic inheritance exists. So one question is, for example, what are the evolutionary origins of the mechanisms behind epigenetic inheritance? How did this temporally extended plasticity evolve? That's a good question, because we have to think about the genetics of epigenetics in the same way that we want to think about the genetics of cultural evolution or anything. But that's not the only question, of course, that we want to answer. But it's an important question and legitimate. The other question we want to ask, and we can discuss it if you want in the Q&A. And then we can ask, well, under what condition is transgenerational epigenetic inheritance advantageous? Why should such a thing develop? And there are many answers to that. It's advantageous in many different conditions. And again, we can discuss it later, but that's not a problem for traditional modern synthesis kind of thinking, because it is recognized that epigenetic inheritance is a developmental mechanism, and yes, it can be transmitted a little bit from one generation to the next. Now, what are the problems that people are having? Why is it difficult for them to accept that when we think about evolution, we should think about epigenetic inheritance? So, are there any interesting consequences to epigenetic inheritance? So I want to list a few, and again, I don't have enough time to talk about all of them, but I will just list a few and then give you a few examples for each of them. First of all, epigenetic inheritance has effects on genetic evolution, on DNA. Two, it affects the dynamics of phenotypic change in populations, even if there is no genetic change. Third, it contributes directly to adaptations, to some adaptations, quite interesting adaptations. 4, it contributes to macroevolution. And 5, it has influenced most of the major evolutionary transition in evolution. All the big changes in evolution had an epigenetic aspect to them. I don't think I can talk about all these aspects. I have written about it with Marian quite a lot, and in Evolution in 4 Dimensions, we discussed this, especially in the new edition, which unfortunately I think has not been translated into Spanish. But it's the same as the old edition, except there is one big chapter at the end. That's the only difference. So if somebody has the patience to translate it, it would be nice. But anyway. So, okay, so let's look at the effects on genetic evolution. First, I will start with the last one because it's the simplest. The simplest effect is that the effect of epigenetic variations on mutability. So, for example, methylated cytosine is 4 to 15 times more mutable than non-methylated cytosine, right? So when we change methylation patterns by inducing a change, developmental change, we change mutability. And there's also very heavily methylated regions, which are called, which usually are heterochromatic regions, tend to undergo less recombination than non-methylated regions. And this is just DNA methylation, about which we know, as I said, quite a lot, relatively a lot. So it can have an effect on, direct effect on mutability of DNA. And when we're thinking about genetic changes, we have to take this into consideration, obviously. Now, the other thing that I want to give you an example is that when we methylate, for example, an allele, silencing through, for example, DNA methylation can mask dominant deleterious mutations. And this can change the dynamics of evolution, of selection, obviously. It can also, by the way, uncover recessive beneficial mutations. So there are all kinds of very interesting effects on the dynamics of genetic variations if we take into consideration the epigenetics, right? That's a very simple thing. Also, this, by the way, wouldn't worry the people who are thinking about the modern synthesis too much. A little bit, because it complicates life, but not too much. Now, epigenetic population dynamics. Well, since at least in plants, we can have long-term epigenetic variation, epigenetic inheritance. By the way, usually the environment doesn't just change from one generation to the next. You know, if you have, we have climate change, for example, and the environment becomes hot, it doesn't suddenly, immediately after one generation become cold, unfortunately. Yeah, it goes on being hot for many, many, many generations, and it induces the same epigenetic variations again and again. And in addition to that, even if there are fluctuations in the temperature, they will be inherited. So I'm not saying this because of theoretical reasons, but because experiments have been made to show that this is indeed the case, again, in the famous Arabidopsis thaliana. So the lab of Collot, Vincent Collot in France, in Paris, they did a lot of experiments. They're working very hard on this, and what they showed is, I'm reading to you, several differentially methylated regions, which are called DMRs, act as bona fide epigenetic quantitative trait loci, accounting from 60 to 90% of the heritability for two complex traits, flowering time and primary root length. Now, these two traits are, of course, important, you know, they have survival value in different environments, flowering time, root length, yeah? And the heritability of these things in pure lines, there is 60 to 90% of heritability to them, which is due to heritable variations in methylation, in DNA methylation. And they say many of the experimentally induced DMRs are also variable in natural population of this species and thus provide an epigenetic basis for Darwinian evolution, independently of DNA sequence change. Okay? It's a very, very good paper which was published in Science in 2014. And there are more papers like that. So we have, it is clear that selection is going on at the level of epigenetics, even when there are no variations, genetic variations, or very, very few. You never have zero, because such things don't exist. Now, of course, in most cases, you have both genetic and epigenetic variations. It's very difficult to tease them apart. That is why people are working on pure lines and on parthenogenetic species and so on. Not because this is the most important thing, but because this is the only way to tease apart the two types of variations and to show, first of all, that this thing exists and that it is important and that selection can occur at this level. Now, in addition to that, we have some population epigenetics models. And there are many, many of them. Here are just, I'm just showing you some examples. All of these models show in different ways that with epigenetic inheritance, population dynamics change both at the epigenetic and the genetic level. And, you know, we need these models in the same way that we needed them in order to show that genetic variation is important in natural populations. That it is really that genetic variation is really important. In the same way, we need models in order to, you know, clarify our thoughts about epigenetic variation and the way that epigenetic variation and genetic variation are connected and how they can influence each other and so on and so forth. There are models that cover quite a lot of the possibilities. And they usually make very conservative assumptions, like, you know, 3 or 4 generations maximum of epigenetic inheritance. And this is enough to change completely the dynamics, the dynamics of a population dynamics. Now, I said that there are specific adaptations that are based on epigenetic inheritance. For example, cellular immunity in C. elegans, in the worm C. elegans, is mediated by RNAi systems. Virus silencing small RNAs are inherited. So there is a whole a type of adaptation that is based on epigenetic systems. We also know, for example, that duplication and insertions in the fungi, neurospora, in the soma and the germline are heritably silenced. And it's a defense against foreign or selfish DNA. And this is an epigenetic system. They're silenced by DNA methylation. Again, I'm talking a lot about DNA methylation, but I don't want you to misunderstand me. It's not because it is the most important. It is because it is the most researched, except for small RNAs, which are also very, very well researched. We know quite a lot about histone modifications, but not enough. And there can be something that Yoav Swen called adaptive improvisation. So what happens is that if you sort of impose stress, a new stress, completely new stress on an organism, a stress that it has never seen in its evolutionary history, the first thing that an organism does is it's not waiting for mutations to happen. It's trying to cope, and it's trying to cope by exploring. So it's trying to change its development in all kinds of ways. Because if it will wait for mutations, it will die for sure. Sometimes it doesn't manage. Many times it doesn't manage. It tries very hard, and there are all kinds of processes that can be recruited to adjust the organism to a new environment. As I say, they sometimes fail. Often they fail, but there is some chance. And there are experiments that show that under completely new conditions, such processes of exploration within developmental explorations are happening, that once some kind of solution is found, it is stabilized, and it is inherited through epigenetic mechanisms. This was found both in yeast and in Drosophila melanogaster, in the fruit fly. Now, I said that there are contributions to evolution and to macroevolution. For example, there can be prezygotic, of course, prezygotic isolation. It will be the same as genetic prezygotic isolation, but it will be much more rapidly established through environmental induction in different environments. And there could be also post-zygotic isolation that results from chromatin incompatibility. So if you induce variations in two populations, or they occur randomly, even epigenetic ones, and they will occur very quickly because epigenetic variation accumulate very rapidly, then the two populations can be incompatible. And I will not go to the third one. And I don't want to go through this, but if we go through each of the major evolutionary transitions, for example, the ones listed by Satmary and John Maynard Smith, we can see that in each case, there was a contribution of epigenetic mechanisms were recruited in order to deal with this, were recruited in order to deal and shape the transition. The other thing that epigenetics is very important for is if we're trying to understand, it's another tool that we have which we can use in order to understand how population change over time. So we can study, for example, not only ancient DNA, but ancient methylation patterns in DNA. And we can compare, for example, the methylomes, the methylation patterns in Neanderthals, Denisovan, and present-day Homo sapiens. And this has been done, and it has been shown that there are important variations, especially in genes that affect certain diseases in humans and affect the development of the brain. So comparative epigenomics of not in general, and including comparative epigenomics in ancient DNA or chromatin, will enrich phylogenetic studies, and we will understand much better the history, not only of our species, but of other species as well. But our species is quite interesting for us, so obviously we want to, we can uncover many interesting things from the patterns of gene expression or chromatin, that something that is related much more closely, patterns of gene expression, such as the methylation patterns of DNA. So that's another thing that we can use in order to understand evolution. So a lot of aspects of evolution can be greatly influenced by what we learn about by taking epigenetics seriously and understanding it. Now, when we're talking about the extended evolutionary synthesis, now many of these things are in the making. Yes, people are studying now population epigenetics. People are studying methylomes in ancient DNA. People are studying the effects of, are studying population epigenetics in theoretical population epigenetics, as well as field population epigenetics. People are doing all these things. People are studying the changes that occur during speciation. For example, in hybridization, what is happening is huge epigenetic changes. People are studying what is happening during the domestication process. And we know that methylation is enormously important in this process. So we are accumulating data about the importance of epigenetics in all kinds of evolutionary processes, microevolution and macroevolution. Now, I think that, and this is only when we are looking at epigenetics in I'm looking I was focusing on I was not talking about culture. I was not talking about the transmission of behaviors. I was not talking about the interaction between the transmission of behaviors and the underlying epigenetics, which is very interesting and very important. I was not talking about all this, but all this are subject of intense study today, and there are very, very good groups that are working on it. And the genetic, the epigenetic, the behavioral, non-symbolic, and the symbolic variations, all of them, when we think about evolution, we have to take them into consideration. Of course, symbolic variations are not terribly important for flies, but they're very important for humans. So we have to think about what to include, not to include. But obviously, when we're thinking about human evolution, we have to think about all these dimensions of heredity. And it will simply not do to think about, to ignore them. We shall have a very impoverished view of evolution. And this is one of the things that the extended evolutionary synthesis is saying. Look, we have to enrich and extend the way that we think about evolution. So let's go back to what Ray said. Ray said that the modern synthesis already incorporated most of the new findings. This is not true. Me and my colleagues, we looked at 10 textbooks, the most used textbooks in evolution, and the kind of thing that we're talking about, the epigenetic inheritance, plasticity, niche construction, are marginalized or non-existent, or relegated to little boxes. This is what is happening. This is a fact. Now, the second point that he had was that the list of subjects that need to be incorporated and given center stage is ad hoc. It's not ad hoc. In all cases, there is a focus on development and a gestalt switch that I was mentioning. You think about phenotype first. Genes are followers in evolution. They are not the leaders. At first, you make a phenotypic change, and then the genes that support this change are also selected. But you start with the phenotype, not with the genotype. Not that the genotype is not important, of course it is, but genetic variations follow phenotypic variations rather than, in most cases, not all cases, of course, but in most cases. The third thing is that the data coming from the study of non-genetic inheritance, especially epigenetics, is not strong enough to warrant a change. And all I could say is simply, this is not the case. It does warrant a change. There is a lot of data about the existence of epigenetic inheritance everywhere. There's a lot of theoretical studies showing how important it must be, even with very conservative assumptions. And we have enough studies already to suggest that it does warrant change. So I think that the extended evolutionary synthesis makes a difference to and so it is where we need a change. And this change will make a difference to biological, philosophical, and methodological aspects of modern biology. Now, I don't have much time, but if I have 5 minutes or 6 minutes, I will go through some of those. Do you want me? Okay. So, I want to go through some biological theoretical claims, then I will go to reformulate okay. The biological theoretical claims are the following. First of all, it's a developmental system approach to heredity and evolution. This is the first thing. This is a system approach. It begins with system dynamics, with a phenotype, which is a developmental system. The agency of the organism is central to this view. The organism is not passive. It is not just sitting there and mutations happen. It is acting in the environment. It is trying to cope. Phenotypic variations, heritable genotypic and heritable phenotypic variations can be decoupled. It's not the case that every phenotypic variation needs to have corresponding genotypic variation. We can have the same genotype with several different phenotypes which are heritable. Phenotypic variations are rarely produced randomly, even if many genetic variations are produced randomly. The phenotypic variations are not. They are usually a response to some kind of conditions. It's not a simple response. Maybe that there is a lot of stochasticity there, but at the developmental level there is a lot of stochasticity at the molecular level and the cellular level. But at the end, there is a coherent physiological adaptive response, and it is not random. It is the result of all this coping mechanism that West Eberhard called phenotypic accommodation mechanisms. Inheritance is based on both replicative and reconstructive processes. I already talked a little bit about it, so I will not go on. Constructive and transformative Lamarckian processes interact with selection at different levels. Okay, so we have the fact that something is induced by the environment doesn't mean that it is not afterwards selected. What can be induced? There can be a range of things that are induced, some of them more adaptive and some of them less adaptive, right? But there will be there will be some directionality. The stochasticity is not absolute. It's much more limited with these systems. Horizontal gene transfers, genome fusions, and gen sharing through hybridogenesis and symbiosis is far more common than was once thought, and can lead to saltational changes and lead to some web-like phylogenetic patterns. Selection is very important. In fact, it has a more expanded domain, because now we have to think not only about selection of genes, but selection that include internal processes within the organism, and epigenetic variations, and behavioral variations, and cultural variations. So in a way, the role of our notion of selection has to be broader. Selection also includes exploration and selective stabilization without replication, for example, in the nervous system. And there is no privileged level of evolutionary analysis. We don't have to start with the genes. In many cases, it is much better to start with the phenotype, with the reconstructed heritable phenotype, phenotypic variations. So these are quite important theoretical claims. And if you agree with these claims, then this is a different point of view that we have about how we should think about evolution. Now, this is a kind of picture that we tried to condense a lot of this stuff. We, again, is me and Marian in a very, very small and new book, which is called, here it is, yeah. It's, once in my lifetime, I managed to write a short book. So most of the books that I write are fat. This is the book. It's a nice book, and it's called Inherited Systems and the Extended Evolutionary Synthesis. And basically what I'm talking about here is what we did in this book, more or less. But very, very, of course, much more. Although it's a small book, we had more space there. So what we see here, all that we start with something, with, let's say, a gamete with DNA, or with an embryo with DNA, with RNA, and with DNA and with cytoplasm, and with non-DNA parts to it. And this little organism responds to changes in the environment. As a result of it, it changes or constructs its niche. This leads to changes in conditions that induce epigenetic modification. It can change its behavior. The response of the organism can be affected by and also affect its microbiome. There will be selection within the organism and among cells, and there will be phenotypic accommodation. Then all kinds there can be a horizontal gene transfer, mutation, and recombination within this organism, which will affect its DNA and can affect its gametes. But within all the changes, the behavioral modification, change conditions that induce epigenetic modification, and so on, can go to the gametes by affecting chromatin marks, small RNA, prions, the frequency selection of organelles within the cell. And this can all affect the profile of the gamete. And then we get to generation N+1. So if we think about evolution, and in addition to that, we have transmission, which is not through the gametes. We have the microbiota is transmitted from soma to soma. Behavior is transmitted from soma to soma. The niche is transmitted through the activities of the organisms. So we have both somatic and gametic inheritance of both genetic and non-genetic variations. And the nature of the N+1 generation depends on all of these factors, not only what happens to the little black blob. Now, there are all kinds of ways of philosophical implications for it. I will not go into it. I want to go to the social and methodological issues that are interesting. First of all, what does it matter? How is the whole way that we think and practice science changed as a result of epigenetics? How is the practice of science changed? So first of all, we have to extend mathematical and simulation models in order to incorporate this. It's a very simple change, but it is a theoretical change that we have to do. We have to have new model organisms. We use clonal organism or pathogenic organism in order to study this aspect of heredity. So we have new model organisms now. We have to have new model organisms. We have to develop new techniques for teasing apart different inheritance inputs. For example, there are already developed CRISPR-based methods for manipulating methylation patterns, for changing methylation patterns, not only DNA, and ways of figuring DNA. So once we have epigenetics, we have to develop new techniques to deal with it. Laboratories and influential people are beginning to change. So first of all, old people die, and some of them retire and stop promoting the old dogmas. And the assessment of ecological, medical, ecological, social importance of extended heredity and evolution is becoming very common. We see it. I mean, we see new institutions, new labs that are dealing with these things. Popular books and professional books are being published about the subject. And also a lot of, there are new journals all the time that are appearing, which are dealing with all kinds of aspects of epigenetics, and I have a big list of those. And there is a lot of hype as well. A lot of nonsense is talked about, is being promoted by all kinds of charlatans. Normal thing, the same happened with genetics. But it is part of this sociology of it. And if you are looking at it, you are seeing that there is a change in practice, and a big change. And all these things, if you are reading Cohn, and you are looking at what he defined as a paradigm or disciplinary matrix, it's exactly this thing that he was talking about. He, and before him, Ludwig Fleck, a great sociologist of science. Very interesting. So what does it mean? Does it mean that all we need is a cosmetic change to the modern synthesis? What would count as a significant revision? So I will just finish with what Marin and I suggested. Well, it's very difficult to say when you're inside this process of change what it means. But let me tell you what we think. We give an analogy. You know, in the 1960s, there was a kind of something called the cognitive revolution, from behaviorism to cognitivism. Now, behaviorism was focused on input. Yes, you had a stimulus, you had a response, and you had the reinforcement in the middle. And what happened all the processes that happened in the middle, all the affordances, all the constraints didn't exist. came cognitivism and said, this is the important story. The insight, these algorithms that are shaping the response, shaping how the processing aspects of how the input is processed and so on and so on. This is what's important. And this was called the cognitive revolution. We think that this is very similar. What is happening is rather similar to this. With modern synthesis, you have a mutation, you have a phenotype, and you have a selection process. Very similar to the stimulus response reinforcement story. What happens? The developmental constraints, all the development is something that is complicating life basically, but it's not this thing that we want to study. Whereas with the ES, the extended evolutionary synthesis, it's very, very central. So we think that it is rather similar to this, and we think that it is a big change in orientation. It requires a gestalt switch. We don't have to throw away what we have learned. On the contrary, we embrace it, but we have to think about it differently and study it and study evolution in additional ways. Now, does it mean that it will change quickly? I'm not sure. I've been around for quite a while, as you can see. Yes, and I want to cite Tversky, who was Kahneman and Tversky. He didn't get the Nobel Prize because he died, unfortunately. System 1 and System 2, he was one of the two people who developed it. And he said, theories are not refuted, they're embarrassed. And people can live with embarrassment for a very long time. So how long it will take for the paradigm to shift, I don't know, but I think it will. Thank you very much.
Well, thank you, Eva. Thanks for the presentation. It was very clear. I am quite convinced of the importance of epigenetic inherence and evolution. And I think that you give us a clear picture about the complexity of the evolutionary theory in the actuality and with the current knowledge. I will do my best to incorporate all this vision in future years of our evolutionary classes. And I have no questions. For me, the three last slides were very clear. And so I open the space for the students and the public.
Thank you. I'm not the Pope, so don't take me too seriously.
Amen.
Okay, well, thank you so much for the opportunity. It's not the first time that I meet you, Eva. We were in 2015 in Mexico, in IPv2, I think. So it's a pleasure to see you again, talking about these topics. I wanted to ask you, well, I have like 4 questions, but I think it's too much. I'm gonna focus only on 2. The first one is this discussion of an extension of synthesis is not new. Even in times of previous, in the 19th, at the end of the 19th, Gould published a paper talking about the extension of the synthesis. And Lewontin, as a critic to the extension, not in the way as the current critics today, like keeping the modern synthesis alive, Lewontin was a critic much more interesting. He said, why we are talking about extension and not a new kind of synthesis? And he said, well, I'm not sure if he said, there is a discussion interesting, but I think that is very similar to the physics situation. Why a single equation for the world? Why a single law? Why we are thinking in a single theoretical framework for evolution? If virus has different evolution to bacteria, bacteria have different evolution to eukaryote. That's the first question.
Okay, so there are really 2 questions. First of all, why is it an extension and not a revision, for example, right? And why not have a pluralistic view of evolution? This is two questions. So the first question is, you're right. I mean, Marian and I call it the extended evolutionary synthesis because this is what has become common. I think it's more than an extension. I think it's a gestalt switch, as I said. It's a kind of different way of thinking about it. So I agree that I'm more radical than the extended. So that's the first answer. The second answer is that I think that we want that pluralism is very good, but we also want to find the common threads that unite the living world. And there are such common threads. For example, what I said about epigenetic inheritance. Epigenetic inheritance is not exactly the same. Not all organisms have the same mechanisms, but all organisms have epigenetic inheritance without exception. That's very interesting. And I think once you start thinking about it, it's also obvious why this is the case. So I think that on the one hand, we have to be very sensitive to the fact that we have different mechanisms in different organisms. For example, C. elegans, like Drosophila, doesn't have DNA methylation, but it has very, very nice transmission of small RNAs. It has an enzyme that replicates small RNAs. It's a kind of parallel transmission of replication system to the replication of DNA. And it has very, very nice inheritance of histone modifications. So we have to be sensitive to these differences and to the way that different epigenetic mechanisms are recruited in different organisms. And we shall have different evolutionary stories to tell about them. But we do want also to find a general way. When I'm saying, let's think about the phenotype first, take a phenotype first view of evolution, I'm not necessarily saying that this is always the case. No, but I do say that this is most likely to be the case in most cases. Okay? So I am making a generalization, and this kind of generalizations, we want them in science, as well as the sensitivity to specific cases, to specific groups of organisms.
Wonderful. And the last is also citing a work of Gould, but not actually Gould. It's a book from Kim Sterrenly, in a book that he writes, Dawkins versus Gould. And I want to be fair with the quote. He shared a quote that I see again in your talk today. You were saying genes are not leads, are followers. It's very similar quote to what Kim Sterrenly think that maybe was one of the conclusions that Gould was wanted for, but he was not geneticist, so he couldn't say what he actually what Kim Sterrenly say in his book. I put the quote in the chat to be completely fair with it. I can read it. Gould thinks gene difference do not cause evolutionary change in population. They register those changes. Do you think that's the change that also do you want with the extension of evolutionary synthesis?
Well, first of all, I want to tell you that the idea that genes are followers, not leaders in evolution, which is what Mary Jane West Eberhard wrote in her book in 2003, was also said by Lloyd Morgan in the late 19th century. Almost in these words, not exactly in these words, but very, very similar. He said, first come the phenotype, and then the genes are selected that support it. That's what he said. So we have this idea around for a very, very long time. It was not Gould, everybody who is a developmental biologist understands it. And everybody who sees how organisms cope with the environment. Now, with Gould, I think Gould had very good intuitions. I think that in many cases, when he was arguing for example, Dawkins, they were talking past each other very much a lot of the time. and they were talking about different things quite a lot of the time. What interested Dawkins was heredity, and he looked at heredity, and he still looks at heredity as the heredity of genes. For him, epigenetic inheritance is not important. It's just something that happens from time to time, but it really isn't important for anything for evolutionary thought. But he's interested in heredity, whereas what Gould was interested was in patterns of evolution. And I think that they were talking from such different points of view that a lot of this of the discussion was a discussion like between two deaf people. I don't know if this answers your question, but I just want to, for me, what is important to stress is that the idea of genes as followers has been around for at least 120 years, more.
Thank you.
I have a question. So my feeling was that the modern synthesis had a lot of success. It was able to frame a whole set of ideas and concepts and processes within a mathematical framework. Do you think incorporating the ES framework into the picture will be possible with using a similar kind of maths? And do you think if that's not the case or not so easy, that might be a reason why it's rejected by many people by modern synthesis?
No, I don't think so. I think that the mathematical models, first of all, are done. There are all kinds of models. One of them is taking the price equations and extending it for epigenetic cases. That has been done by several people, more than one. There are population epigenetic models that are not very complicated. I mean, there's no problem with having a mathematical framework. I mean, if you really think about the mathematical framework of Fisher and Holden and Wright, I mean, it wasn't very complicated, for God's sake. I mean, we teach it in population genetics, right? I mean, it's not a complicated thing. Everybody can understand it. And I mean, yes, you can complicate it, of course. But usually you don't have more than 3 genes. And the one epistatic interaction is already too much. So, it has very little to do with reality, right? And now, it's not the only mathematical models that we have. There are great models that are using molecular evolution, that are not based on the Hardy-Weinberg equilibrium, right? And are not necessarily interested in Hardy-Weinberg equilibrium either. They're very good models. And of course you can make these models for epigenetics. It's not a problem at all. The problem is not this. If people, one of the things I hope somebody will do, I don't want to do it, but I hope somebody will do one day, is take all the models that already exist and put them together in a book of population epigenetics, so that there will be one book that people can look at and say, look, these are the models that already exist, and there are enough models, they exist. I hope somebody will do it one day. It will be a nice book, and also will show where we need to work. So always these books show you where the lacunas are. So this is something that I hope will happen sooner or later. I have been promoting this among my students, but none of them did it, unfortunately. But I hope maybe here I'm talking to people, maybe somebody will put all these things together. It's not too much work, really. And write something about it and put it, and so that people can actually consult such a book. I don't think this is a problem at all. I think the problem is sociological. It's the internal and external sociology of science. This is what the problem is. I don't think it's a scientific problem. I don't think it's a theoretical problem. I think it's a sociological problem, political problem, to some extent. Political in the sociological sense.
Okay, thanks.
Related with that, Lican Martinez is asking in the chat, what evidence do you think are needed to be found to produce a robust and definitive paradigm shift towards the ES? And well, I believe that you answered most of the question.
Well, I'll tell you the story, okay? One day I talked with a young colleague of mine, and I asked her how, what to do, what should we do in order to make a robust change in the way that people think about epigenetics. And she looked at me and said, Eva, you're a dinosaur. And she said, this is, of course, I mean, everybody knows it. It's just, we need more evidence, we need to do more experiments, we need to have more models. It's normal science now. Yeah. But I said, in my world, I still have to fight for this. She said, yes, because you're old. So maybe this is the question, the answer.
If I can interrupt with that part, I'm following classes on epigenetics, and it's completely true what you're saying. It's not new for many of them. However, they do not talk about evolution. Like when you talk with them, hey, that interesting explanation of methylation, but do you know how can impact the evolution of the organisms that you are studying? No, well, that topic is complicated and it's hard to study. It's very hard to study the inheritance, incomplete inheritance, things that could happen in this generation and affect not the next one, but the next one. Like in humans, woman can carry 2 generations ahead. That's something very complicated in his own body in 1 generation, affecting 2 generations ahead. So technical limitations are still affecting the young research in epigenetic, molecular epigenetic.
I think there are lots of, there's a lot to do. You're right, and a lot of people who are doing molecular biology, they don't think like evolutionary biologists. What we need is more evolutionary biologists who think about epigenetics. There are not many of them. And this is a pity. The reason that I'm talking to you here is that I'm one of the few people with Marian that actually started to talk about it very seriously in the 1980s, many, many years ago, when most of you were not born. So that's the reason, yeah, because and still it is the case that a lot of people are not talking too much about it. There are people, they are beginning to, the number of people who are doing evolutionary epigenetics is growing, but it's growing slowly because, evolutionary, what they're interested in when they're thinking about epigenetics is medical issues, is developmental issues. These are the burning issues for people. Evolutionary biology, okay. And evolutionary biology is dominated by population geneticists. And they have been socialized in a very different world, and they don't know epigenetics. Many, many of them are very, very ignorant. I'm very sorry to say. And the combination of taking taking a complex evolutionary view and putting it together with epigenetics is not something that many people are engaged with, more than there were. Yes, I mean, once upon a time, there weren't very many. A friend of mine asked me, how was it to walk in the desert, to be a lone voice in the desert? I said, it was very nice. It was only me and Marian in the desert. It was quiet. It was no problems. But, now, thank God, it's good. There are more people, but not enough. So that's why your teachers, I mean, do they know evolutionary biology? Most of the molecular biologists, they don't.
Okay, I believe that Ariel wants to ask a question.
Yes. Hi, Eva. Been a fan for a while now.
Yes, my question was, do you think epigenomics and epigenetic diversity can be used as a species conservation tool in the face of global warming? And what are the obstacles, the hurdles that we need to overcome in order to apply it?
Yeah, I think it's a very good question. And the answer is, I really don't know, but it's something that we have to test. This is exactly the kind of studies, this is exactly one of the things that when we go to the field and we try to see how different species are coping with global warming, one of the things we want to see is how they react genetically and epigenetically. And it could be a good diagnostic tool. But I don't know that it will be. It may be.
Okay, Maria Cecilia Mestre in the YouTube channel is asking, hi, nice talk. I wonder about time scale. Epigenetics seems closer in time, while classical genetic ignorance seems to go further back. How this work?
Closer in time, you mean that the rate of change is faster? I'm not sure I understand.
I think that she's asking about the how many generation that this change can be
inherited
or have impact. Yes.
Okay, it depends on the species, right? So, for example, there was a paper in Arabidopsis and in many plants. It can be, if you are looking at the DMRs, at the differentially methylated regions, we're talking no, single sites even. You are talking about 5 to the minus 10 to the minus 4. So 1 in 5,000, something like that. Which is, you know, you have mutations with this rate in some repetitive elements. This is the rate of mutation of some repetitive elements. So this is quite, so this can be inherited for 100,000 generations. Then you have for example, in C. elegans, people were talking about 14 generations of inheritance in a recent paper. In Drosophila, I remember a paper talking about 50 generations. In mammals, usually people are talking 3 generations, in some cases 6 generations, depends. With difficult to study mammals, but because of long generation times and such things. But these are the usual. Sometimes it could be more, it could be longer. It also depends what exactly you are looking for, what exactly you are looking at. Then you have, you know, it's also, I think, variable in the sense that it could be the case that if you have persistent induction of epigenetic variation, generation after generation after generation, it can lead to very, very stable epigenetic marks, because it can extend, for example, how widespread the methylation pattern is, which can induce this, can stabilize its reconstruction. Things like that can happen. We don't know enough. What we know is that the range of variations is variable, very variable, and taxon dependent, it seems.
I have one more question. Years ago, I saw a presentation in a congress that showed that mutation in bacteria in different media under different stressful conditions had bias. And you showed that mutability is altered by epigenetic change. Do you believe that there is a way for organisms to increase the probability of mutation and to accelerate the evolution in some conditions?
I don't think so. I think that there is evidence for this. And Barbara McClintock suggested it. We know that under stress, transposable elements jump more.
Mm-hmm.
This is increasing the rate of variation in the genome. Under stress, for example, if you take Drosophila and you put it into heat stress during a particular developmental period, right? You suddenly have recombination in chromosome 4. Chromosome 4 usually doesn't undergo any recombination at all under normal conditions. Under stress, heat stress, it does. We know that stress changes recombination, changes the rate of mutation, changes transposition. We know these things. And it makes a lot of sense. If you are under stress, and it is an unpredictable stress, not something that you know how to cope with. The only way to deal with the unknown is by throwing the unknown at it. Yeah. So you play some kind, you gamble. It's a gamble. And if you are able to gamble, at least you have a chance. If you don't gamble, you have no chance, you will die. So this is one of the ways in which organisms cope with the environment. This is what you do when you don't have any other way. This is the most radical way of coping, and it's very, very widespread. And there is a lot of evidence for it.
Okay, thanks. Any more
We
questions?
have a couple more questions from the YouTube chat. Carmela Lucia 40 is asking if it's well known how epigenetic transmission is achieved between cell lines, and where she can get some information about that.
There is a lot of information about, well, a lot. We know quite a lot about DNA methylation again. In cell lines, in somatic cell lines. In what is happening in germ cells is less known, less well known. We begin to understand it more, but it's very complicated. And it's, as I say, it's not simple. It's not a simple replication process. It's a reconstruction process. That's why it's so complicated. So, but in cell lines, yes, somatic cell lines, yes, there is quite a lot. There's quite a lot of information in some organisms about transmission of small RNAs. Again, a lot of gaps in our knowledge, but we're beginning to know a lot more than we did just 5 years ago. So there is huge advances there, but still there's a lot we don't know.
Thanks. And then Mika Soategi is asking, what do you think has been the impact of your fascinating book, Evolution in 4 Dimensions, the publication to date?
I'm the last one to know. You know, I really don't know. All I can tell you is that we were very surprised that such a fat book was translated into so many languages. That was, and I think that we were very lucky in that it came at the right time. That it was a time where people were more ready to think about it than before. So it's sort of because the work on epigenetics was exploding during this period in the beginning of the 2000s. Like 20 years ago, it was beginning to really explode all over the place. And people were also doing a lot of more work on animal traditions and niche construction, and some important books appeared, and cultural evolution was getting a lot of, I mean, it started before, but at that time there was a conjunction of a lot of interest in these things. And I think epigenetic in 4 dimensions put it together in a systematic way, in a way that that was not too difficult for people to understand. We tried very much to write it in an accessible style and with as little jargon as possible. So I think that this was helpful. And what we see, what Marian and I see, is that the people that it is affecting young people, and it is affecting people not only from biology, but from sociology and from psychology and from anthropology, and even political science, believe it or not, and all kinds of people from a lot of. It makes sense. Also, I think it makes, you see yourself that identical twins are not the same person. Yes, they're different from each other in many, many ways. You see it with your own eyes. You can't believe that genetics is the whole story. You yourself, in your life, you encounter things that suggest that Dawkins cannot be right, that there is more to the process of evolution and heredity than what he suggests. And it's a common sense in a way. And I think this common sense actually found an ear during this particular period. But I think it was the right timing. So I don't really know what our contribution was to the change in outlook that is beginning to happen, because it is beginning to happen. I'm not sure, but I hope that it did something, that we're part of the people that helped to, because I think that this is a better way of looking at things, and is more has something to do with reality.
Well, it certainly changed I think there are grants that have been given to study EES that wouldn't have been given 30 years ago. So I think it made a huge impact. It opened up the field.
Well, it's one of the things, there were several things that, and several things that came together. I mean, our studies and, as I said, cultural evolution studies and studies of behavior and studies of and niche construction studies, all these things sort of came together, and a lot of, of course, studies on developmental plasticity. I think the importance of development became very, very obvious.
Thanks.
Any more questions?
If it's possible, just a question about an update. In 2015, you made a talk about social epigenetics, and I remember you were using the same metaphor of the Waddington landscape, but not for epigenetics or for the epigenotype, but you were talking something like social opportunities. There is a social landscape of opportunities, but we can actively manage it. And that's why you all at the end mentioned it's impacting also politics. Yes, thanks to you. You put that topics on that context. How is that work?
Yeah, we developed a kind of way of thinking about cultural evolution, not as an analogy, genetic evolution, but to take the epigenetic landscape of Waddington as a metaphor, and then use this metaphor to create another metaphor, a metaphor of kind of social landscape, where there are many, many different forces that shape this landscape, and people biographies are shaped. They are like the people's lives are sort of going to this cultural attractor that is formed by all these forces, and they contribute to the reconstruction of the social system. And we gave some examples, for example, urban poverty in the United States, which is a self-sustaining system because of bad schools, because of bad food, because of alcoholism, because of because of drugs, because of violence, because of people because people who are poor tend to live in kind of ghettos or favelas or areas that are theirs, and people who manage to get out of this don't come back and don't contribute to the community. And so there is a self-sustaining loop that is being formed there. And this loop you can think about it as a kind of network or as an attractor that is part of a network. And we thought that this kind of thing, and when you're thinking in this way, what you're thinking about is really the feedbacks between different aspects of the social system. Some of them will be epigenetics. For example, if you're eating all kinds of horrible things and drinking alcohol and when you're pregnant, God forbids. By the way, I'm telling the men, you also shouldn't do it. Mm-hmm. It's not good for the offspring. Or taking drugs, or doing all kinds of, this affects the epigenetic profile of the offspring. And unless drastic things are done in order to change this, and it is possible to change it, it is not impossible, but usually it will not be changed because there is nothing to change, because there's no effort to change it seriously. And you can't change just one thing. Good schools are not enough. I mean, you have to change the way the parents behave. You have to change you have to change many things at the same time. It's a complex system. So we thought that if you identify in this complex system the places where there is positive feedback, and if you change them, it will affect many other places within this network. This will be helpful. So we were talking about so when I was talking about social epigenetics, I was talking about this kind of picture in which epigenetics is part of the story. It's not the whole story. Of course not. It's part of the story. It is, the molecular correlate, a developmental correlate of these things. So, and we have to think about it and to understand that when we are polluting the environment, we're creating poverty, we're creating chronicle wars and all this kind of things, we're affecting future generations in a very, very real physiological sense. This is what we're doing. And we have to take responsibility. I mean, we all have to take responsibility for it, especially politicians, of course, who don't know what epigenetics is, never heard about it. And it's time they did. And it's part of the world in which we live. I mean, we live many people live in a toxic world. And this toxic world is not just their world. It's the world of their children and grandchildren, if nothing is done to change it. And we have to change it in a very proactive way. It's not just enough to stop. We have to invest in changing it, because we're introducing real changes into the epigenetics of the next generations.
Thanks. Well, Eva, thanks again. We know that it's late.
It's all right. I'm in London. It's 8:49.
Okay. It was a pleasure to have you as a guest. And I have to say that I loved your book, Evolution in 4 Dimensions, and I hope to read your new book soon. And
it's a very different thing.
Okay. Well, for me, it was an honor.
Which one?
The last one, the inherent extended evolutionary synthesis.
That's all right. That's a good. It's a useful little book. It's really, yeah.
Okay. So thanks again, and
thank you, and good luck. It was a pleasure. Bye.
Un saludo también a todos los asistentes. Los esperamos el miércoles que viene con la charla de Robert Pennock.
Thank you very much. Bye bye.
Thanks.
Bye bye.